Welcome to Click for IB Biology. We're studying genetics and we're going to find out in this video how to perform a test cross in dihybrid genetics. There's a blank version of this presentation, a PDF, is linked into the description below, where you'll also find a quiz of 10 questions with answers so that you can determine how much you've learned from this particular topic. So let's remind ourselves what the purpose of a test cross actually is. With a heterozygote, it has the same phenotype as the homozygote dominant. Now, to illustrate that, here we have an F1 pea plant which has a green pod, which is the dominant phenotype for the pod, and it has a yellow seed, and that's the dominant phenotype for the seed color. Now we use the genotype for this particular specimen is big G, big Y, little y. So let's just go over here and do big G, big Y, little y. Now realize that the homozygous dominant would be big G, big Y, big Y. But we can't tell the difference between those two with the actual plants. So not knowing what the genotype of the specimen is, we can't tell whether it's heterozygous or whether it's homozygous dominant. And so the solution to our problem is to carry out a dihybrid test cross. And we do that by crossing with the double recessive specimen, little g, little y, showing the yellow pod and the green seed, both recessive characteristics. Now, if we work through the combinations in meiosis, we have to separate our homologous pairs, but we have to do so independently, meaning that from this particular specimen, we would get big G, big Y, big G, little y, little g, big Y, and then we would get little g, little y. But from this one, we're only going to get little g, little y. So when we work through those combinations, we get big G, big Y, little y, big G, little y, small g, big Y, little y, and finally little g, little y, little y. Now let's think about the phenotypes that we would get from that. So this particular individual, this particular fertilization, that would end up being green. So that's a green pod with a yellow seed. Now this one would be green pod but with a green seed. On the next level with little g, so we're now on yellow pods. And we have the big Y means yellow seed, which is dominant. And then we have the double recessive, in which case we have the yellow pod and the green seed. And notice, and green seed. And so that's our phenotype. These are our genotypes. And notice that they come out in the ratio of 1 to 1 to 1, like this. So if we have a heterozygote for 2 genes in the test cross with the double recessive, our outcome would be 1 to 1 to 1 to 1 in the phenotype. If we were to perform the same test with a homozygous dominant, then we would only get one phenotype coming out of this cross, which would not have the ratio, the test cross ratio of 1 to 1 to 1. So that's the test cross ratio, one to look out for in questions. Okay, So now we move on to the next genetics video. There's a chance to subscribe to the site. That's always greatly appreciated. And there's the entire playlist linked in for you for the genetics videos.